got to where I am. But yeah, so I was born and raised in New York City, which often surprises a lot of the people that I work with, because you don't often find too many people from big cities ending up working in wildlife, because we don't have as much exposure to like nature around us or opportunities to explore the outdoors. But I was always interested in the wildlife around me, even in the city. Whether it was watching the gulls on the river or even the rats on the subway. But I didn't realize that doing this type of stuff, like just observing the natural world, could be an actual job or career until I got to college. And so I went to college not too far from home in central New York at Cornell University. And my first science research experience was working with fruit flies in the lab. And I really loved working there because I got to work with so many cool, I got to work with so many cool people, and had some really great mentors, including these two, Dr. Brian Lazaro and Dr. Mariah Chambers, who helped me design my own experiments and do my own work. And the nice thing about working with Drosophila, as I know some of you work on fruit flies, is that you can have a lot of experiments fail without too much consequence, which is great for an undergrad who is still trying to figure out how to do this type of work. And so it was a lot of fun, but during my time, I realized that I really wanted to pursue biology research long-term, but I didn't just want to continue working like only in a lab. And so after I graduated, I actually went back home to New York City and worked for a year at Columbia University as a technician in the lab, specifically Dr. Dustin Rubenstein's lab. But this position also had a field component all the way on the other side of the world in Kenya, and I got to go there and assist with field research there. And so I assisted a graduate student at the time, Sarah Gindry Parker, who is now actually a professor at Kennesaw State University. But I got to work really closely with her studying superb starlings, which was my first time working with birds. And during that time, I just absolutely fell in love with these birds and working with birds. I was working on mainly a breeding study. And so here I have, I'm holding just a couple of few-day-old baby chicks as we were checking the nest and getting blood samples from them. And after this, I just knew I wanted to continue working with birds. So during this year, I was also applying for graduate school, specifically PhD programs, where I would be able to do some type of wildlife biology research. And so now I'm here in New Orleans as a graduate student at Tulane. I'm in the 6th year of my program in Dr. Jordan Karubian's lab, which is kind of crazy to say that I'm in my 6th year. And I'm also defending my dissertation in just over a month, which is also crazy to say, and something I'm just gonna not try to think about too much right now and move past. But one of the focuses of my lab is studying ornamentation in animals. And so when we look at ornaments across taxa, we see this huge diversity of signals in nature. Ornaments can have really important consequences for individual fitness and reproductive success via intra and intersexual interactions. And subsequent studies have revealed the importance of ornaments on the evolution of biodiversity through speciation processes. And we can really see this clearly in birds, where ornamentation in color is one of the most conspicuous traits to differ among closely related species and often evolves under strong sexual selection. But the genomic, endocrine, and physiological factors that determine ornamentation are often studied independently of each other, leading to this incomplete understanding of how underlying forces interact to produce and maintain these traits. And so for my dissertation, I've been using an integrative approach to study the evolution and mechanisms of the production of a plumage sexual signal in this species, the Red-Backed Fairywren. So these small songbirds are found in Australia and have been a really useful system to study the production of plumage ornamentation at both an intra and inter-population scale for a few reasons. So first, within a population, males exhibit flexible reproductive phenotypes, where some males express this ornamented red-black plumage right here, where the red feathers in this red feather patch are produced using carotenoid pigments, and other males exhibit female-like unornamented brown plumage. Both types of males can breed, but there's evidence that the red plumage patch is a sexually selected trait. So first of all, the red plumage patch is used in courtship displays, and only males with red feathers will court females. And so to give you all an idea of what that actually looks like, I'm going to play a video where here we have two ornamented males and several females down here. And so this male really puffs up those back feathers and then does a quick little somersault. And I'm going to play this again in slow motion, but you can see how this male really is pushing those feathers up over his whole body and just like shoving it in the faces of these females, really trying to show them off. And then he'll just do a little like somersault over them, which is really cute. But here I'm also going to play another quick video of an example of courtship. So this all happens really fast, but here again we have these two males, and then out of focus right here is a female. This male starts pushing those feathers up over his back and then does a somersault again in front of the female, trying to really show off those red feathers. And so plumage type does have reproductive consequences, so that courtship is good for something because ornamented males have higher reproductive success than unornamented males. And in addition, previous work has found that the androgen testosterone is really important for signal acquisition. So ornamented males have higher levels of circulating testosterone than unornamented males, and testosterone levels increase as males molt from brown plumage to red-black plumage. However, there's still this black box here of how on a mechanistic level does testosterone control this really ecologically relevant phenotype. So I hypothesize that testosterone must be inducing gene expression to produce these different plumage types. And so my first objective was to figure out what genes are differentially expressed for an individual to transition from unornamented to ornamented plumage. So we can also zoom out from this intrapopulation differences where ornamented red-backed fairywren males vary between two geographic populations with two recognized subspecies that differ in plumage. So we have our crimson population, our crimson subspecies to the west, and then our orange subspecies to the east, with a relatively narrow contact zone where the redder plumage has digressed across. And so my second objective is to understand what genetic mechanisms underlie the subspecies differences, as well as the introgression of plumage color. So the first thing I wanted to know was whether these different male types had different types or levels of the pigment that is used to produce that orange and red coloration. And so these pigments, like I mentioned briefly, are called carotenoids. Carotenoids are actually the pigment that is used to produce most of the red, orange, or yellow colors that we see in vertebrates. And so to look to see if there were differences in the level of carotenoids circulating, I went out into the field and caught a bunch of red-backed fairywren. So I caught unornamented males like this guy here, ornamented males and females as well. I would catch them and then collected a small blood sample. So this is what it looks like if you haven't seen how many of us collect blood from birds, but we poke their brachial vein, which is basically like in their armpit with a small needle, and then we collect blood in a capillary tube. Then I centrifuged that blood down and separated the plasma, which is where the carotenoid pigments are, from the red blood cells. And this is what that looks like. So here, every tube is an individual blood sample from like a different individual. And this is just the plasma. And you can see even just by looking at it head on that there's already variation in plasma color. So probably this more yellow plasma belonged to like a female or an unornamented male. And then this really dark, almost like bright red probably belonged to an ornamented male. And there's a lot of variation between kind of these two ends. So after I collected these samples, we ran HPLC on them, which is high performance liquid chromatography. And this not only enables us to figure out what the concentration of carotenoids are in the sample, but also the types of carotenoids, because there are like 200 different types of carotenoids out there. So the first thing we found was that there are actually 6 different carotenoids found in the plasma of these birds, 2 dietary. So these are the carotenoids that the fairy wrens get directly from the food they eat, and they tend to be more yellow in color. And then we had 4 ketocarotenoids, which are metabolized after being ingested, and those tend to be more orange or red in color. And so what we found was that ketocarotenoid concentration was associated with sex and plumage. So specifically, red-backed fairywren ornamented males had much higher levels of carotenoids than unornamented or females. And so the next thing I wanted to figure out was what's leading to these differences in carotenoid concentration? And my hunch was not only that gene expression was involved, but specifically this one gene, CYP2J19, was involved. And that's because this gene is a known carotenoid ketolase, which basically means that it metabolizes those dietary carotenoids into ketocarotenoids, which are, again, more orange or red. So to test this hypothesis, I went back out into the field and caught red-backed fairywrens and implanted unornamented males with a small beeswax implant that released biologically relevant levels of testosterone. So this T implant induced those males to start molting into ornamented plumage. And at the same time as we did those T implants, I had also implanted more unornamented males, but with a sham implant, so a beeswax implant with no testosterone as a control. And 10 to 12 days later after implantation, as those T implanted males were starting to molt, we collected both the T implant birds, the sham implant birds, but also naturally ornamented males as well as females. And after some really intense days of fieldwork, this was some of the most intense fieldwork I had done, which involved crossing rivers while carrying a centrifuge and a cooler full of dry ice and like tables and stuff, getting up before sunrise to try to set up everything to catch the birds before the sun went up, which is when the birds were most active. This is my colleague and collaborator, Dr. Hubert Schwabel, who's a professor at Washington State University, sitting in our table in the very, very dark early morning, and also just dealing with more mosquitoes than is just okay. Here, every little dot is a mosquito that you can see. I'm wearing this rain jacket mainly because of the mosquitoes. It wasn't raining, but the mosquitoes were so awful out there, and there are even more that's just covering my back that you can't see. But anyway, after all this, and I got these samples back home, I ran qPCR, which measures relative gene expression, and found that CYP2J19 is more highly expressed in the liver of ornamented males than control unornamented males. So here is this difference there. And if you're wondering why I picked the liver, it's because it's known to be an important site for carotenoid metabolism. And even more compelling, testosterone implanted males had higher CYP2J19 expression than control males and were at the same level of expression as naturally ornamented males. And so these results suggest that testosterone upregulates CYP2J19 expression and that this expression is associated with plumage. And so this is, to our knowledge, the first time that hormonal regulation of a specific locus had been linked to carotenoid production in natural context, which can reveal how endocrine mechanisms produce sexual signals that shape reproductive success. And me and my colleagues actually published this study last year in Proceedings B, so it's really exciting to have this work out there. But this was just one gene, right? Which we can probably safely assume that there are probably multiple genes responsible for this plumage production. And so to take this a step further, I conducted a transcriptomic study using these same samples. So as a reminder of my study design, I had collected samples from females, control unornamented males, T-implanted unornamented males, and ornamented red-black males. Like I had mentioned, I had collected liver samples, liver tissue, but if you're interested in plumage production, it probably makes sense to also look at what genes are being expressed in those feathers. And so I collected growing back feathers, the color of those shown in the boxes, just as they were erupting from the sheath. And then I sequenced the transcriptome of both these sets of samples, aligned the sequences to our annotated Red-Backed Fairywren reference genome, and ran a differential expression analysis. And so to back up a little, I was not only interested in which genes are differentially expressed between plumage phenotypes, but specifically which genes testosterone is regulating. And so to visualize this, I created this plot where the x-axis is the log fold change of expression between unornamented and ornamented males, where genes on the right, so that would be in this area, or more highly expressed in unornamented males. And then similarly on the y-axis is the log fold change between unornamented males and T-implant males. And so genes that would be in the top right quadrant will be those that are shared upregulated between these two comparisons, suggesting that testosterone is important for mediating expression of these genes associated with difference in phenotypes, so specifically having that red plumage. And then the bottom left is similar, but for those associated with brown plumage. And so here I have shared differential expression in the liver, where every dot here represents a gene, and the ones in red are those that were significant in both comparisons. And so there are only two genes in that top right quadrant. This one, which I've been talking about for the last 10 minutes, is CYP2J19, the same gene that I ran the qPCR experiment on. And so this was expected to see this gene up here, but it was also nice to see that the RNA sequencing data validated the qPCR data. But the second gene here is ELOBL6, which stands for elongation of very long fatty acids. And this is really interesting because carotenoids actually have really similar structure to fatty acids and lipids. And so fatty acid genes may also be involved in carotenoid processing. So now when we look at the genes for the back feathers, we generally see more genes differentially expressed, but several of those are associated with transport functions, which I have circled here in black. And these two starred ones are specifically involved with lipid transport. And again, carotenoids have similar structures to lipids. So these may actually be important for moving carotenoids into feathers in our fairywrens. And so altogether, these data suggest that testosterone does differentially regulate the expression of possible carotenoid genes in different tissues associated with plumage color. So it's cool to see how this one hormone is able to change expression differently in different tissues to produce plumage. So now I'm going to move on to my second objective. And I had previously mentioned that there are two distinct subspecies of red-backed fairy-wren with a narrow contact zone. Hybridization is extensive in the system, where hybrids exhibit variation in the hue of their red plumage patch across and between the natural range of either pure subspecies. And hybrid zones can be really useful for studying phenotypic variation. Because when two species, or in my case, subspecies, interbreed, it often results in offspring with intermediate appearances, which you can see here in these tanagers and in these orioles. And hybrids provide a powerful window into how new species and new traits evolve. And hybrid zones serve as this natural laboratory for discovering the genetic basis for traits like coloration. So not only is there an existing hybrid zone with variation in plumage color in red-backed fairywrens, previous work has also found that first, there's experimental evidence that females prefer to mate with redder males over orange ones, and redder males sire more extra pair young. Second, clinal analysis suggests that this female preference is probably leading to the introgression of redder plumage across the hybrid zone, where the plumage line is discordant from the genetic line. And third, using a reduced representation genetic data set, red and orange individuals are genetically indistinguishable in the hybrid zone. And so this provides a really powerful system in which to study the genetic mechanisms that control the sexually selected color trait. Because if we sample birds from within the hybrid zone, genetic differentiation should only be linked to coloration and not to fixation of neutral regions or local adaptation that's common between allopatric populations. And so I sampled from the middle of the hybrid zone, and I sequenced whole genomes of the 10 most red and 10 most orange birds from this 15-year data set we had of 332 unique males. And I also sequenced birds from allopatric populations as a control comparison and to test for introgression. I aligned my sequences to our reference genome. I recovered around 26 million SNPs with an average of 10x coverage per individual. I also wanted to just take a moment to say that this work was done in collaboration with lots of folks at the Cornell Lab of Ornithology, and especially these two, Dr. Jen Walsh and Dr. Irby Lovett. And I really would not have been able to do this genomic work without them. But so after I collected my data, I ran a PCA on the SNP data to see how similar these groups of birds were to each other. And as expected, all individuals in the hybrid zone were indistinguishable from each other, even when their plumage does differ, but allopatric populations cluster separately. I then estimated FST across the genome in 25 kb windows, first comparing the red and orange hybrid zone birds to each other. And so this is a Manhattan plot. And for those unfamiliar with this type of plot, on the x-axis here, I have chromosomes. Each dot represents a 25 kb window of genetic data. And then on the y-axis is FST, which is a measure of genetic differentiation. So if your FST is 1, that means the two groups you're comparing are completely different from each other. And if the FST is 0, that means they're completely the same. But note here in this plot, my FST only goes up to 0.2. So though divergence is very low overall, the global FST in this group is pretty much 0. There are still some small peaks that show up in a few places. So to validate these peaks, I also estimated FST for my 2 allopatric populations. And as expected, there are many peaks, but some of those were probably driven to fixation by chance. And but however, peaks that are shared between these 2 comparisons should be linked to color. And this is how I chose the candidate peaks that are putatively linked to color variation, which I've highlighted here in blue. And so I wanted to see if there are any functions that were shared by the genes in these elevated regions to see if there are any functions that might be related to plumage color differences. And so I ran an enrichment analysis on the genes in all these elevated regions. And so in brief, this type of analysis compares my list of genes that I give it to all the described genes in the chicken genome, and then groups the associated gene ontology terms by how similar they are, and then produces this gene map, or sorry, this tree map, which with the most enriched functions coming out proportionally by size. And so the first big function that pops out is this hormone-mediated signaling pathway, which is interesting because, like I've talked about before, we know that testosterone is important for signal acquisition. So it's cool to see these functions pop up in the genomic data as well. And a second big function that pops up is fatty acid synthesis, which, like I've mentioned many times now, is really interesting because carotenoids, those red or orange pigments, have really similar structures to fatty acids and lipids. So those type of fatty acid genes may also have carotenoid coloration functions in birds. So while I was able to find some interesting functions that were shared among genes in these peaks, I also wanted to see if I could zoom in closer to find specific loci associated with plumage color across the species range, not just in these few populations. So after I had identified those peaks, I developed primers to sequence 120 300 base pair amplicons in those peaks that contain SNPs that were fixed in the allopatric birds and still diverged in the hybrid zone birds. So I amplotyped 285 individuals across the 16 populations shown on this map as a transect, with the goal being to find SNPs that correlated with color across the whole species range. So after sequencing and filtering, I retained 85 amplicons and ran a PCA on these data where I colored the points here by hue value, which is how red or orange the birds are. And we can start to see a general pattern where PC1, which can be thought of as a genotype score for individuals, seems to track hue, which is what we would predict given how I designed these primers. But to better assess this relationship, I also plotted the PC1 score on the x-axis to the hue score on the y and ran a linear model and found that this relationship is significant. But my main goal was to see if I could find individual SNPs that were concordant with the introgressed plumage cline, which would suggest a link between these loci and the observed red plumage introgression here. So in this plot, on the x-axis, I have distance along the transect, and each one of these points is represents a population along the transect. And we have the neutral genetic cline in black, which can be thought of as a hybrid index. And then the plumage cline is in red, where values at 1 are the most crimson color and values at 0 are the most orange. And you can see that these clines are discordant. And then in blue, I've overlaid the individual SNP clines from my amplicon data. And out of my 85 amplicons, around 50% of them had SNPs concordant with the plumage cline, and therefore may be candidates for coloration. And so one of these SNPs, whose cline is shown here in blue, is in the gene FASN, which is a fatty acid synthase, which again is a gene of interest because of how carotenoid pigments have similar structures to fatty acids. And as you can see, this SNP seems to have introgressed alongside the plumage cline and alongside that plumage introgression. And so this gene may be involved with color differences in red-backed fairywrens. I also compared the individual genotype of the SNP on the x-axis here to the hue value on the y, and you can clearly see a relationship there. And when I run a linear model, again, the R-squared value is almost 0.2, which means that this one SNP explains 20% of the variance in individual color, which is quite high for a single SNP. And so taken all together, this suggests that the snip and fasten in part underlies color variation in the species and may be the proximate mechanism for the plumage introgression. And so integrating all this information on the role of hormones and gene expression and interpopulation flexible phenotypes with that on the genomic mechanisms of introgression on a species-wide scale can help us understand how new traits come about and the processes that mediate the evolution of sexual signals. So for example, here I found that the genes responsible for within population color differences are not the same as the ones for between species or subspecies rather, which suggests that, there are probably complicated mechanisms at play. But more generally, I think that this really highlights the power of using an integrative approach to understand color evolution, which can be applied to many different systems. And so with that, that's the end of the research portion of my talk, but I just wanted to spend a minute talking about something that I've been thinking about a lot, sort of throughout my whole PhD, as I've been doing my work, but especially now as I'm coming to the end of my program and reflecting a lot about the work I've been doing. But so a lot of people say that we've entered the age of the omics, right? So genomics, transcriptomics, proteomics, there's many more than that as well. And I think many of us that work with these tools acknowledge that we're probably well past entered that. It's sort of become the standard of science. And the omics work that I do on these birds, I do to further understand this really ecologically relevant trait, right? Because we know that this plumage trait is important for reproductive success and is moving through populations. But we wouldn't know that, I wouldn't know that if other scientists before me didn't spend many years rigorously taking note of what's happening in the system in the wild. And yes, working with wild systems can be a little frustrating at times. It can be difficult when you start trying to get to the nitty-gritty of how things work, which is a lot of what my own work has been. And I've complained to colleagues and friends about how I can't keep fairywrens in captivity. These birds just don't do well in captivity, in part because they hold large territories, they're really social, and live in these social groups. They're insectivores, which just makes it really difficult to feed them. Because if I could keep them in captivity, there would be so many cool experiments I could do with them. But I've also come to appreciate how important this natural history is, not only to my work, but in general, especially as it seems like this type of work, this natural history work, has become devalued in our field. And so because I cannot bring these birds into the lab, that means I need to do more field work to answer my questions, and I'm definitely okay with that. And what's exciting is that I will be able to do more field work with them after my PhD, because I'm really excited to say that I will get to continue working with them in my postdoc. So I received an NSF postdoc research fellowship, and I'll be starting back at Cornell actually, but more closely with the Cornell Lab of Ornithology in March 2022. I'm going to continue asking questions about plumage production, red-backed fairywrens, but I'm also going to leverage that long-term data set that I had mentioned briefly about to try to connect this plumage variation to reproductive success in the hybrid zone, also to test for heritability of color. And so with that, I wanted to thank just so many people that have helped me along the way, my many field techs who I could not have collected all this data without, so many friends and colleagues in Australia that help us work out there and get all our paperwork set for that. and lots of people that have helped me in the lab and with analyzing my data. And Jessica French, who illustrated these really, I think, beautiful images of the two red-backed fairywren subspecies. And yeah, and with that, I guess I'll take any questions.